Hi, let's discuss about medical imaging in biomedical engineering. So this time we want to study about current biomedical imaging technology, and there are various items. So first, I want to show by Vesalius this diagram. He was a physician from Belgium, and in this 16th century, you can see he has made this human brain dissection. This is based on a real dissection. So one can realize inside the brain, there is this ventricle. Without opening it, we couldn't have understood this before. And majority of the biomedical imaging can start with an image. So the image is started from a photography. This is the first photography in 1827 in France. And the B bottom is a modern photography. So these photography or started the biomedical imaging, we can consider in this way. So how biomedical imaging has revolutionized medicine and biology. So our eyes, our retina can only recognize on the surface of the body in most of the time. But however, biomedical imaging allows us to see inside our body and to visualize our biological structure and even functions down to the microscopic scale. So what is image? Image is a representation of measurable properties that vary with spatial position. And oftentimes means if we have multiple images which is taken at different time points, then we can start to understand the dynamic nature of the subject. So image can provide detailed information about biological first as the space, so this is structures, okay? And powerful imaging modalities also provide information, for example, molecular, cellular, and biophysical process, which we mean it as a function. So conventional imaging, we talk about structure, but more powerful imaging, we also want to see the underlying processes that means its function of the biological structures. So, and methods to visualize and quantify these properties from microscopic to macroscopic levels are now becoming available. So image information can be used for clinically for disease, yes, diagnosis, figuring out what kind of disease it is, we can confirm, and for monitoring of a treatment. So even we can use imaging information for monitoring whether the treatment has been taken properly, as well as scientifically for normal and abnormal structure and physiology. So imaging is becoming the most popular and powerful technology we are using for modern medicine. So let me just start with a new imaging technology example. So first, like gene expression imaging. Have you ever thought about this? Based on spatial radioisotopes, which is coupled with genetic methods. So even using radioisotope coupled with genetic methods, we can even visualize gene expression in imaging. So such an example is in advanced PET or positron emission tomography. And the second is a real-time brain imaging during cognitive tasks to guide a neurosurgery. So during neurosurgery, such as, let's say, resection of brain tumor, you don't want to resect a very critical functional part of the brain. So you give the patient to do a relevant cognitive task to visualize the part of the brain. And that technology is called, yes, functional MRI. So, and also right after a heart attack, a patient went right into the clinic and you want to see the action of the heart to identify the injuries. And to map that mechanical action of the heart, with a specialized ultrasound, which we call, because it's hard, so it's called cardiac ultrasound. I will discuss more about this later. So these imaging technologies are essential tools in medicine because they provide a spatial map, enabling our physicians to localize the biological phenomena being examined in space and time. I just have 3 example pictures here. So the left part is a brilliant, very detailed map of inside the body made by X-ray computed tomography, or CT. And second one, this is a black and white, but this is an example of fMRI. Usually based on this, you can find a location that is lighting up for the localized neural activity. And the third is a little poorer spatial resolution. However, it provides a metabolic activity of the brain, which is done by a PET, positron emission tomography. So I'm going to start to discuss about methods to create an image. So to create an image, we need first an energy source, okay? So such as electrons, ultrasound for ultrasonic imaging, light for optical imaging, X-rays for X-ray, CT, or radio frequency for magnetic resonance imaging. And that energy source will interact with the target, for example, like human body parts or organs or cells, and that produces a signal. So I brought one example here. You may be familiar with, you can look at this. This is an example of ultrasonic imaging. And even there's overlaying this red and blue color. This is more functional aspects, such as of blood flow. So this ultrasound, there's energy source is ultrasound. You send it through this transducer, which send the sound wave, and those interface in mechanical structures will bounce off layer by layer, and that is detected by the same transducer, which is converting sound wave into unmeasurable form, such as voltage. So that is a transducer, converting one form of an energy to another form of a signal, okay? So oftentimes, it may be necessary to introduce some kind of material into the subject to highlight a particular structure or function. For example, the barium sulfate for CT. And I brought a picture of here, that the contrast material. So barium sulfate enema makes these large intestine to be visualized with CT so that one can find out if there's any problem in the gut. So here I want to, there's a big busy table that biomedical imaging invention history. So you can see from very long time, almost over 400 years ago, starting with the microscopy. In 1827, I showed an example of photography, and 1896 X-ray gives a 2D projected structure by Röntgen, and let me see, ultrasound and confocal microscope and gamma camera and SPECT and computed tomography from 1972 and by this Hounsfield and Cormack, and PET 1973 by Michael Phelps, and 1973 MRI technique showing 3D structure and function by Lauterbur and Mansfield. And you see these rectangular ones. What do I mean by this? This is actually showing Nobel laureate. So many of these biomedical imaging inventions, in fact, led to a big contribution to our medicine, so that it can even get a multiple Nobel Prize. So at the last, I want to bring you a big table which compares these various biomedical imaging modalities. And so this is a busy slide. So, but by the end of all this lecture, you may come back to this to understand the various aspects. So CT, computed tomography, magnetic resonance imaging, and single photon emission computed tomography, SPECT or positron emission tomography, PET, and ultrasound has all different penetration level. CT and SPECT, PET can penetrate organ tissue, and you can see SPECT can even see tissue cellular level, while their spatial resolution is depending on the energy source, and the cost is also depending on the instrumentation and the number and types of detector. And fundamentally, it's important to understand what are the electromagnetic radiation spectrum, which is the energy source. For CT, it's X-rays, and MRI, it's radio waves. SPECT and PET uses these gamma rays, which is higher energy than X-ray. And ultrasound uses high frequency sound. So this is very inert for our body. And advantages of CT is it can show bone, excuse me, and anatomical imaging as very well. While MRI shows very good soft tissue contrast, SPECT and PET has a relatively limited spatial resolution. However, these are more functional imaging, especially PET. You can see the sensitivity is very high, okay? And while ultrasound is very low cost, so this is one of the most abundant medical devices in the clinic, and it provides a real-time imaging. And while disadvantage as a CT, SPECT, and PET are using X-ray and gamma ray, which are ionizing radiation, which can be harmful potentially. So we have to be careful about those. So radiation, radiation, radiation. While CT is very good at bone and contrast agent, while it has a poor soft tissue contrast. In comparison with the MRI, having very good soft tissue contrast. So while MRI is costly and the time for scanning is a little longer, like tens of minutes. SPECT and PET has a limited spatial resolution, which I will discuss later. And ultrasound, its resolution depends on the frequency of this signal source and transducer, but in general, it's relatively limited spatial resolution. Okay, thank you for your attention.